We're going to talk about the biology of mind and behavior. If you don't have a background in biology or haven't really learned this before, it can be a little bit overwhelming. Plus, the chapter is a little bit long, so I'm going to, my treatment of some of this stuff will probably be a little superficial. But here we go. There's the brain, I guess, which is kind of the structural aspects of the different lobes, and then there's the mind. And they appear to exist independent from each other. This thing, the mind, our ability to remember and plan, our consciousness, it emerges from the brain, but in the end it's hard to reduce it to the brain. But in the end, again, they're not distinct. The brain results in the mind, and I think one day, we're going to understand how the mind actually maps onto the brain fully. But we're nowhere close to that now. The basic kind of building block of the brain and the central nervous system is the neuron. This is, a very small unit. In fact, there's like 100 billion neurons in the brain. These neurons receive signals from the sense organs or from other neurons. They process the signals, and then oftentimes they send the signals onto other neurons or muscles or organs. All thought, feeling, and behavior is reduced to the firing of neurons. The complexity of the human brain is really mind-boggling. If we have 100 billion neurons, and on average each neuron connects to 10,000 of other neurons, or have 10,000 connections, we're talking about 10 trillion neural connections, which is, I can't even get my mind around that. Here's an example of a neuron. The cell membrane is the outside. The cell body is kind of like the brain, the nucleus. The axons are what transmit the kind of firing of a neuron down the axon, which tends to be covered with this kind of myelin sheath, which we'll talk about later, to the terminal boutons. And the terminal boutons are almost touching but not touching other neurons. Instead, they secrete a neurotransmitter substance that goes into this sort of synaptic cleft and influences the other neurons. And the dendrites are what receive information from other neurons. Here's a picture of kind of what one could look like and how it branches off and whatnot. Now, the firing of a neuron is a little bit complicated. Let me just say this, that it's an electrochemical event. And when the neuron isn't firing, it's more positively charged on the outside than the inside. And when a certain threshold is reached, the neuron fires in this kind of all or nothing fashion. And when it fires, it allows different ions to flow through channels and membranes, and it conducts down the axon, something called saltatory conduction, kind of leaping from myelin sheath to myelin sheath. I'm not going to really go into much more detail than that, other than the myelin, which you see around the axon, not only acts as kind of an insulator to the axon, but allows these signals to travel more efficiently, and much more sort of energy efficiently and fast. And in some disorders that are very common, like multiple sclerosis, reflect the deterioration in the myelin, and you can see how it affects our ability to think and behave. Now, what you're seeing here is, if we were to measure the change in the kind of membrane potential or the voltage when a neuron fires, and above you'll see that different ions, whether it be sodium or potassium, are traveling in or outside of the neuron. The best way to look at it, though, is first let's take a look at the threshold of excitation. What you see then is a very rapid reversal where the neuron goes from negative 70 millivolts to positive 40 millivolts. Then it shoots back down very quickly, dips below negative 70 millivolts, and then comes to rest at 70 millivolts. I want you to look this over closely in your notes, because what's happening is as sodium is entering the cell and potassium is leaving the cell, it's these positively charged ions that are causing this electrochemical change, or this electrical change. Then, when you get to the synaptic cleft in the terminal boutons, this is at the end of the axon, you have the synapse. The cleft is that little area before, in between where it touches the dendrite. Then you have various neurotransmitters inside the terminal bouton in these little vesicles, which break when the action potential, the firing neuron, triggers into. And what happens is the neurochemicals go into the synaptic cleft, they bind to the dendrite, and it either excites or inhibits, depending on the various kinds of sort of dendrites and receptors on the other end. And this is sort of what they call the synaptic transmission. Now, certain drugs that we can take act like, in a sense, kind of act like neurotransmitters. And sometimes they serve as an agonist, sometimes as an antagonist. So something like serotonin or dopamine, acetylcholine, or epinephrine, these are agonists. They act like, maybe they facilitate the neurotransmitter, or act like the neurotransmitter. Popular ones are these selective serotonin reuptake inhibitors. And I think I forgot to mention in the previous slide that when there's leftover neurotransmitter, oftentimes it gets taken back up into the terminal bouton. In most cases, I think with acetylcholine, it actually gets destroyed. But the point is that it's reused. Some of the common antidepressants are selective serotonin reuptake inhibitors. They inhibit the reuptake of the serotonin. It stays in the synaptic cleft longer, and it tends to make us feel better, like Prozac and Zoloft and Paxil and things like that. And so now, remember, the neurons are the building blocks. When we think of the central nervous system, we think of everything from the brain through the spinal cord. And now we're going to spend a little time kind of talking about the spinal cord. And as you can see there, it goes through the back. It's protected by these vertebrae, and in between these vertebrae you have neurons branching out and innervating all parts of our bodies. And it tends to be, the vertebrae or the spine tends to be kind of divided between the cervical, thoracic, lumbar, and sacral. And as you can, if you have time to read, you can see that paralysis occurs at different points depending on the break. The higher the break, the more paralysis over more of the body. The lower the break, the less paralysis. Then the spinal cord actually connects to the brain. The cerebral cortex, that kind of outside convoluted area, the cerebral hemispheres, we have kind of a left and a right. Lobes, which we'll talk about in a minute, so these hemispheres are divided into lobes, and then this thing called the corpus callosum, which facilitates communication between the two hemispheres. Now here are the lobes, and again, you'd have the right hemisphere and left hemisphere. This is looking at the right side of somebody's head. But there's a occipital lobe near the back, a temporal lobe kind of on the side by your ears, a frontal lobe kind of by the front, and then a parietal lobe kind of on the top in between the frontal and the occipital lobes, and they're separated by various sort of sulci, kind of large grooves. Now, when we think about the frontal lobes, we think about a very important part of the brain that's associated with planning, memory as well, engage of a lot of motor processing, which we'll see, a motor strip, reasoning, emotional regulation, our ability to plan and instigate behavior is very, very important. And there's an interesting story of Phineas Gage, who had a railroad accident where I think some sort of railroad tie or something went in and destroyed part of his frontal lobe, and it talks about how and how his behavior changed after he began to recover from that illness. Then there's the occipital lobe in the back of the head. Just think of vision when you think of the occipital lobe and all kinds of visual processing. So you have these connections from your eyes, this kind of optic nerve that goes back to the occipital lobes, which does all this incredible processing that helps you to be able to see and understand what you're seeing. Then you have the temporal lobes, again the side, kind of in front of the ears there, associated with your ability to comprehend language and sounds, but it also is important for storing memories or entering new information into memory, so very, very important. And then finally, the parietal lobes, the top of the brain, helps with your somatosensory processing, which we'll talk about in a little bit, spatial location, attention, and things like that. Very, very important features. As I mentioned before, the frontal lobe has some responsibilities in movement, and if you'll see near the back of the frontal lobe is a primary motor cortex, and depending on where on that cortex you were to touch or stimulate, it would be responsible for the movement of those kind of body parts. Adjacent to that, at kind of near the front of the parietal lobes, are the primary somatosensory cortex. Here, similarly, they're responding to pain and pressure and hot and cold. And again, it kind of mimics, again, the primary motor cortex. As you can see, a lot of neurons devoted to facial movement, which is very complex, and maybe even the hands, less to things like the trunk, which can't really, isn't really involved in a lot of detailed and exquisite behavior. Then underneath that cerebral cortex are all of these subcortical brain areas. The most primitive, the hindbrain, closest in the connection to the spinal cord, has things like the cerebellum and reticular formation, medulla. Then there's a little midbrain. It's kind of tiny. It has certain functions as well, and then the forebrain, which is believed to have, at least from an evolutionary perspective, developed a little bit later. But the thalamus, hypothalamus, pituitary gland, all of these kind of underneath the cerebral cortex. Just to give you an idea of some of the functions, the thalamus is kind of a relay station for the brain. A lot of information from the lower subcortical areas going up to the cerebral cortex. You have the hypothalamus, which is really involved in a lot of species behaviors, keeping warm, eating, sex drive, thirst, all of that. Hippocampus, essential for forming memories. Amygdala, very important for feelings. Basal ganglia, planning and producing movement. Brainstem, primitive part of the brain, like the medulla and pons, basic sleep and arousal. Cerebellum, coordination of activity, of behavior and activity. You damage the cerebellum, it's very, very hard for you to move. How do we know all this stuff about brains? Well, we've studied electrical activity through electroencephalograph and single cell recordings and magnetoencephalograph. More recently, we're able to visualize and see the structure of the brain through computer-assisted tomography and magnetic resonance imaging, and we're also able to visualize the function, the activity of the brain through like positron emission tomography and functional magnetic resonance imaging. All these things help to fill in this picture of what part of the brain is active and how active. We have learned, because of brain damage and seeing what parts of the brain are damaged and how that affects behavior, or either through lesioning or stroke, and even some stimulating the brain to see how stimulation of the brain results in different behaviors or memories or feelings. Now, all of these neurons, and I really don't want to spend too much time talking about this, have in their cell, DNA, their nucleus, cell nucleus, DNA, so every cell, and we have trillions of these cells, and this exists this long paired strand spiraled into this famous double helix that we've come to know and recognize. The DNA in each cell has billions of these base pairs of proteins, and these base pairs are organized into 23 pairs of these chromosomes, half of which you inherit from your mother, half of which you inherit from your father, and these are what become our genes. Combinations of these base pairs on these different chromosomes represent our genes. Again, I don't want to spend too much more time on this, but please look at your note for additional detail. The question is, it's only been recently since we've been able to study the genetic complexity of humans and other species. I think the argument for a long time was that humans would be genetically more complex and superior to other species. Interestingly enough, genetically, the human is no more complex than a mouse, or not even that much more complex than a plant, even, or a fruit fly. So it's not truly or totally a function of genetic complexity as much as what the genes actually allow you to do. And as far as genetic diversity is concerned, I think there was this notion that we are quite different from other species. But if you look genetically, we are quite similar to a chimpanzee, 98.5, and even 85% of overlap in genes between a mouse. So again, amazing how you can have very similar DNA structures and yet a very different appearance. The sum of all of our genes is called our genotype, and the observable structure of behavior, how a behavior looks and behaves, is called the phenotype. It's the expression of those genes, and these genes are mutating. And in fact, sometimes we can cause the mutations. Sometimes the mutations are random over time or selected by the environment. We've been able to create through mutations different types of fat mice or thin mice. But I think the other important thing to know is that the environment matters too. So oftentimes with these, our brain and our kind of neural connections and whatnot, they become, if we don't use these functions, we lose them over time. They become pruned. And if we are developing other parts of our brains, it grows. This is called plasticity. So the more we practice the piano, or the more we engage in certain things, we have neural growth or neural pruning. But the point is that the neural connections can grow and or diminish. The one thing, of course, DNA is highly heritable, and many of the traits, DNA, you inherit DNA, and there's a great deal of variability in the DNA that you inherit, and it runs in families. So the DNA between identical twins, 100%, between dizygotic twins or brothers and sisters, 50%, and so on and so forth. But this whole idea of behavioral genetics allows us to study how DNA influences behaviors when there are things like identical twins or fraternal twins. And there's the whole kind of evolutionary side to it, the idea that certain genes or certain behaviors, and then as reflected in genes, occur through this kind of natural selection. If we have certain behaviors that are adaptive or that are help the species to reproduce and to be fit, those genes get passed on, and the ones that result in premature death don't. And so you see this kind of evolution of genetic behavior, of genes as well. It's interesting now is that DNA testing is becoming quite quite accessible. In fact, for $985, you can get a genetic blueprint, and there's companies like 23andMe. For just a couple $100, you can find out a lot about your genes. It's of course now used in law. It helps us to understand disease risk and prevention and paternity. But I think we're going to have to watch, because soon, economically, we're going to be able to construct a genetic map for every individual in the United States. And as we learn more about the genes and behavior, I think there's potential for abuses and good things and bad. Discrimination because of certain risk factors or something like that. Okay, I think I'm going to stop here for now. But again, look over your notes. Ask me if you have any questions. This is a fairly complex chapter. Thanks. Bye.